that I wanted to talk about was how we are able to regulate the pyruvate dehydrogenase complex. And there's two mechanisms that we have that play a role in regulation. And one of them is pretty common. I'm sure you're familiarized with, at least at this point, and probably throughout general biology, we talked about negative feedback. So for the negative feedback loops of the pyruvate dehydrogenase complex, you just have to think about what are the products of each and every one of these. But the three ones that I really just two things, and then one's a kind of a phenomenon, would be acetyl-CoA and NADH, and then the energy charge itself is ultimately, in my opinion, a negative feedback loop. So for acetyl-CoA is going to act to inhibit E2. NADH, as you could probably imagine, this is going to be acting to inhibit E3. And then the overall energy charge, I'm just going to go ahead and I guess, talk about this, but the fact that ATP, which is something that ultimately that we're involved in the production of this, is going to act on the pyruvate dehydrogenase complex as a whole to inhibit it. And I'm not saying that this is a direct inhibition, so it's going to be inhibiting it indirectly. Just to make sure that we're not missing the forest for the tree here, the pyruvate dehydrogenase complex is all about making acetyl-CoA, which is all about starting the Krebs cycle, which is all about making electron carriers, which are all about the electron transport chain, which is all about making ATP, right? So ATP has a lot of things that it can inhibit and a lot of things that it's involved with, but if you mess with one step of the reaction, say you mess with glycolysis, you're going to be messing with this, and if you mess with, say, other compounds that we're going to talk about. So ATP and AMP, the energy charge of the cell as a whole, plays a huge role in regulating the entire cell as a whole. The other thing that we have, aside from negative feedback loops, is the process of phosphorylation, because what makes phosphorylation really nice as a means of metabolic control is that we don't really have to wait for a buildup of the concentration of things that we don't need. We can act directly to stop a mechanism or to stop a reaction pathway that's at the time not needed or energetically wasteful. And so, like, for the longest time I got this weird kind of thinking that evolution was all about energy efficiency, and while that's true 90% of the time, it kind of led me to some dangerous attitudes that needed to be changed. But for the purposes of biochemistry, it really does help. Like when you think about how energy efficient our cells are, right? So phosphorylation is something as well. And the energy charge, like I said, it affects everything. That in turn can actually affect whether or not we're going to be phosphorylating certain parts of this. But anyways, this is all happening at the E2. That seems like all I've ever talked about is E2 throughout these two videos. So what we have is, for PDH, a kinase, and then, as you could imagine, if we have a kinase and phosphorylation is a part of our control, the other one would be a phosphatase, and I'll switch colors to draw that. And I hope you're not asking what PDH PDH stands for pyruvate dehydrogenase, in this context, the pyruvate dehydrogenase complex, right? So this is at E2, and this is such a cool, I like to envision it as this two-headed dragon type of thing, because this is all connected to E2, right? So there's got lots of things involved in it. And for the kinase itself, and I think before I kind of mentioned that, I want to talk about what the phosphatase does, but I guess I'll mention the kinase first. For the kinase itself, what it does is it's going to be phosphorylating at E1 to shut off the whole complex. So I just realized that this looks probably confusing to some people. You're thinking, well, wait, you just said phosphorylation at E2. So at E2 we have the Pdh kinase and phosphatase, but the main side of regulation is the very beginning of our reaction, right, which most of that happens in the E1. Phosphorylation to shut off E1. So what are some of the inhibitors of Pdh kinase? Well, ADP, and pyruvate. Okay, notice that I'm listing these as inhibitors of the enzyme Pdh kinase. So Pdh kinase phosphorylates to shut off the E1, right? So if we have a lot of pyruvate, we're obviously going to be inhibiting our Pdh kinase, so we're going to be doing the reaction that pyruvate dehydrogenase complex is going to be doing its job. It's going to be undergoing these reactions that we just talked about. For the Pdh phosphatase, as you can imagine, if phosphorylating something shuts it off, then removing that phosphate, dephosphorylation, is going to be turning it on. So dephosphorylation, the removing of a phosphate group, is going to actually activate E1. So one of the things that I think I had already kind of talked about, but in case you don't remember, is that whereas with kinase reactions, what usually acts as a cofactor is magnesium, for phosphatase reactions, one of the things that activates it, or I think of it just as a cofactor, would be the calcium in the +2 cationic state. So what would be, I guess, signaling terms, something that would activate it? Well, anything that's going to cause the release of cytoplasmic calcium. In some contexts, this can be IP3, which is going to cause to release cytoplasmic calcium. This can be an action potential, which acts on those sarcoplasmic reticulum to release calcium as well through voltage-gated channels. And then lastly, there's other things like hormones that can act to release calcium to act as a cofactor for this reaction.